Namaste students. I am JVN Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic hypersensitivity reactions and the types of the hypersensitivity reactions. What are the hypersensitivity reactions? So we have to understand what is the hypersensitivity and what are the autoimmune. If a immune system is used to cause diseases by itself, and it is an autoimmune disease. And when the immune system is overexpressed and it is not desired, then it is known as the hypersensitivity reactions. We know that immune system has important role in protection and immunes, some cells which are involved in the immune response can bind to the non-self molecules. So when sometimes they identify the self molecule as the non-self molecules and cause disease, then this is the autoimmune disease. And when they bind to some self molecule as a non-self molecule and overexpress the immune system and express the immunity where there is not requirement, then this is the this type of immunity is known as the hypersensitivity reactions. Means there is not requirement of the immune system to respond, but immune system is responding, which leads to the more action of the immune system, which makes the person very sensitized. So this type of activity is known as the hypersensitivity reactions. So hypersensitivity reactions are of four types: type 1, type 2, type 3, and type 4. Type 1 reaction is the IgE mediated, which is the classical allergy type. Type 2 is a IgG and IgM mediated, which cause the lysis of the immune cells. Type 3 is the IgG mediated or complement system based hypersensitivity. And type 4 is a delayed type hypersensitivity. So we will discuss one by one each. First, we discuss about the type of hypersensitivity reactions or classical allergic hypersensitivity. It is mediated by the IgE, which are attached to the mast cells. So a specific antibody will be secreted in the case of the type 1 hypersensitivity, and the symptoms results in the allergic responses, which are known as the anaphylaxis reactions. And the disease include the hay fever, asthma, eczema, bee sting, and the food allergies. And agent, the agent which are involved in the type 1 hypersensitivity are known as the allergens or non-parasite antigens that can stimulate the type 1 hypersensitivity. And these allergens will bind to the Ig and trigger the degranulation of the chemical mediators. These allergens are the low, small molecular weight protein, 15 to 40,000 molecular weight proteins. And what is atopy? Atopy is the term which is used to describe the genetic trait to have a predisposition of the localized anaphylaxis. And atopic individuals have high risk of the type 1 hypersensitivity reactions. So we need to discuss about the mechanism of the allergic reactions. So what is the sensitization? Sensitization is the repeated exposure of allergen initiate immune response that generate Ig isotype and T helper cell number 2, Th2 cell required to provide the IL4, which clashes the Ig. The when we talk about the mechanism of the type 1 hypersensitivity, there are some cytokines which are involved, such as interleukin 4, interleukin 4R, CD40 in the sensitization. Now, in mechanism of sensitivity, the IG will first of all, the allergen will enter into the body, and any kind of dust particle can also act as an allergen. They are not actually the antigen, but the immune system will understand that this is a antigen, and that allergen will react like a antigen to the immune system, and it will be killed. The killing of these allergen is not required, but still immune system is performing the actions to kill that particular kind of antigen. These particles are not harmful, but immune system understand that it is a pathogenic molecule, and they start the response. So first a allergen will enter, and the IG will bind to the mast cells by receptor, which increase the lifespan of the IG. The half-life of IG in the serum is some days, whereas if they are attached to the epsilon receptor, it is increased to the months. The FC receptor and the FC epsilon receptor have high affinity to the IG receptor. Mast cell, basophils, and activated cells are the examples. And allergen first bind to the IG, attacks to the epsilon receptor, which triggers the granules from the cells. First of all, the allergen will come and it will bind to the IG, immunoglobulin E, and immunoglobulin E is attached to the FC epsilon receptor R1, and when it will attach to the bind attached to the IG, it will this FC epsilon R1 receptor will trigger the release of the granules from the mast cells, and these mast cells are attached to the immunoglobulin E. Then this is the first step where the allergen has come on the first time. This is the sensitization. The immune system gets sensitized where it comes in contact with the immunoglobulin E, and the FC epsilon receptor R1, which is bound to that IgE, will start sensitization. It will bind to the mast cells. The mast cells and the pathogen will allergen will expose to the second time. The mast cells will bind on the surface of the IG, and then allergen will bind to the IG and cross link to the activate signal with the tyrosine. And when it will come in contact with the mast cells, it will release some histamine. It will release some histamine, and releasing of that histamine will result to the anaphylaxis reactions. So it was not happened in the first time, but the same allergen will enter into the body and will expose to the mast cells. Then the mast cells will release some chemicals which are known as the histamine, which will go to the anaphylactic reactions, and these anaphylactic reactions will lead to the many unnecessary functions of the body, which are known as the uh, wheezing, diarrhea, vasodilation reaction, and it will also activate the enzymes for the tissue breakdown. The leukotrienes are also the molecules which are secreted by the mast cells. Similarly, prostaglandins are also secreted by the mast cells. Histamine, leukotrienes, and the prostaglandins are secreted. by the mast cells when the pathogen enters in the second time. Some more mediators are also there, which are known as the primary mediators, including the histamine, cytokines, thermonecrophils, factor alpha, interleukin 1, interleukin 6, and enzymes such as tryptase, kinase, uh, cathepsin are also involved in the type 1 uh, immunity, where the primary where they act like a primary mediators, and some secondary mediators are also there, including the leukotrienes, prostaglandins, Th2 cytokines, interleukin 4, interleukin 5, interleukin 13, GM, CSF. What is the role of these interleukins? They have important role in the activation of the mast cells to release histamines. So they activate the mast cells. The sensitization steps may sometimes continue. The mast cells control the immediate response. Eosinophils and neutrophils drive the late chronic pathway, and the eosinophils play key role in the late phase reaction of the type 1 hypersensitivity reactions, and in the sensitization. It makes the prolonged sensitization steps. So some localized anaphylactic reactions are also results. Digestive tract contact results in the vomiting, cramping, diarrhea, skin sensitivity, airway sensitivity, which results in the sneezing, rhinitis, or wheezing, or asthma. So these are the results of the type 1 hypersensitivity reaction. So I hope that students will be able to understand the type 1 hypersensitivity reactions. I will explain the summary here. First of all, an allergen will enter to the body. This allergen will be identified by the some cells which are antigen presenting cells, which will secrete some specific antibody, which are the IgE antibodies. These and IgE antibody will bind to these allergen, and also they will interact to the mast cells. They will bind to the mast cells. This is the sensitization step where the allergen has entered the first time. But when the allergen will enter the second time, then this Ig molecule will come in contact with the mast cells with some receptor Ig receptor family, and they will activate the mast cells. The mast cells will get activated unnecessarily, which cause the mast cells to release histamine and some another molecules which are not necessary to be released. And these histamine will cause the anaphylactic reaction, which may cause the disease like the asthma. So I hope students will get a better understand about the type 1 hypersensitivity reactions. So what kind of questions can be asked? The question can be asked that explain type 1 hypersensitivity reactions. So you can very well explain it. So my lecture is over here. Thank you very much.